Welcome. With this session we start the glaucoma section, and in this session we'll be discussing the relevant anatomy and physiology in glaucoma, and these include aqueous humor formation, aqueous humor drainage, and death of the retinal ganglion cells in the retinal nerve fiber layer and the optic nerve head. The aqueous humor is produced by ciliary processes of the ciliary body in the posterior chamber, and the aqueous humor then travels into the anterior chamber through the pupil and exits from the angle of the eye. A minor fraction of the aqueous humor produced by the ciliary body diffuses into the vitreous cavity. The ciliary body has ciliary processes approximately 80 in number, one of which is represented in this diagram. Each ciliary process has a central core made up of fenestrated capillaries and stroma and is lined by two layers of ciliary epithelium. The layer adjacent to the ciliary stroma is pigmented and is called the pigmented epithelium, and the layer adjacent to the aqueous humor is non-pigmented and is called the non-pigmented epithelium. So this diagram shows a magnified view of the two layers of the ciliary epithelium, the non-pigmented epithelium adjacent to the aqueous and the pigmented epithelium adjacent to the ciliary stroma. Basement membrane of these two epithelial layers are along their basolateral surfaces adjacent to the aqueous humor and the ciliary stroma respectively, and the apical portions of the cells of these two layers face each other. The apical regions of the cells of the non-pigmented epithelium have tight junctions, which are not present between the cells of the pigment epithelial layer. However, gap junctions are present between cells of the two layers as well as between cells of the same layer, and this makes the ciliary epithelium a functional syncytium. Mechanism of aqueous humor production involves three steps. In the first step, solutes and water are transferred from the ciliary stroma to the pigment epithelial cell. In the second step, they are transferred from the pigment epithelial cell to the non-pigmented epithelial cell, and in the third step, they are transferred from the non-pigmented epithelial cell to the aqueous humor. In the first step, the pigment epithelial cell takes up sodium and chloride ions from the ciliary stroma via sodium hydrogen and chloride bicarbonate antipodes. Carbonic anhydrase 2 is present in the cytosol of the pigment epithelial cell, which provides hydrogen and bicarbonate ions required by these antipodes. Carbonic anhydrase enzyme also activates these two antipodes. There is another sympode which transfers sodium, potassium and 2 chloride ions from the ciliary stroma to the pigment epithelial cells. Water enters the pigment epithelial cells through channels following the osmotic gradient created by transfer of these ions from the ciliary stroma to the pigment epithelial cells. In the second step, these ions and water are transferred from the pigment epithelial cell to the non-pigment epithelial cell via gap junctions made up of connexin proteins. In the third step, these ions and water are transferred from the non-pigmented epithelial cell to the aqueous humor, and here sodium-potassium ATPase pump plays a major role. This sodium-potassium ATPase pump transfers 3 sodium ions into the aqueous humor in the anterior chamber in exchange for 2 potassium ions which enter into the non-pigmented epithelial cell. Chloride ions enter the aqueous humor through chloride channels following its electrochemical gradient. There is also another antipode which transfers bicarbonate into the aqueous humor while taking up chloride ion into the non-pigmented epithelial cell. And water follows the osmotic gradient through aquaporin channels, and the rate of exit of water is determined by hydrostatic pressure and osmotic pressure within the capillaries of the ciliary stroma, the intraocular pressure and osmotic pressure of the aqueous. Aqueous humor has 3 routes of exit from the intraocular space. The major ones are the trabecular pathway and the uveoscleral pathway, and a very small amount which passes across the vitreous and the retina into the choroid, which is considered to be important for adhesion of the retina with the retinal pigment epithelium, but contributes little to intraocular pressure. The trabecular pathway is called the pressure sensitive or the conventional pathway, and in this pathway the aqueous humor passes through the trabecular meshwork and the Schlemm's canal to episcleral veins. The uveoscleral pathway is called the pressure insensitive pathway, and in this pathway aqueous humor exits through the root of the iris, the anterior face of the ciliary body between the muscle bundles of the ciliary body into the supraciliary and the suprachoroidal spaces. Trabecular outflow occurs through the trabecular meshwork in the internal scleral sulcus, which is an internal excavation of the corneoscleral junction. So this excavation in the internal face of the sclerocorneal junction is the internal scleral sulcus in which lies the trabecular meshwork. The internal scleral sulcus is limited anteriorly by the Schwalbe's line, which is the peripheral end of the Descemet's membrane, and is limited posteriorly by the scleral spur. The scleral spur is posteriorly attached to the ciliary body, particularly to the longitudinal fibers of the ciliary body, and the scleral spur itself contains myofibroblast-like scleral spur cells oriented circumferentially, which are contractile in nature, and the contraction of both the longitudinal fibers of the ciliary body and the scleral spur cells opens up the trabecular spaces, which increases trabecular outflow. So the internal scleral sulcus contains the trabecular meshwork internally adjacent to the anterior chamber and the Schlemm's canal externally adjacent to the corneoscleral junction. The trabecular meshwork is thus present circumferentially around the globe in the internal scleral sulcus. It is triangular in cross-section with its apex at the Schwalbe's line, and base is continuous with the scleral spur and the ciliary body, and the trabecular meshwork is made up of trabecular beams, which in turn are made up of connective tissues with collagen and elastin lying in the center and basement membrane material lying in the periphery. And these trabecular beams are lined by trabeculocytes, which are endothelial in nature and have phagocytic and contractile properties. And the trabeculocytes are connected to each other by gap junctions and desmosomes, so forming a functional syncytium. So here in this diagram we see the trabeculocytes shown in blue color lining the trabecular beams and connected to each other. From the internal aspect to the external aspect, the trabecular meshwork is divided into three parts: the uveal meshwork shown in brown in this diagram, the corneoscleral meshwork shown in gray in this diagram, and the juxtacanalicular meshwork shown in yellow in this diagram. The uveal meshwork is continuous posteriorly with the iris root and the ciliary body, and the trabecular beams are cord-like and have trabecular apertures larger than those of the corneoscleral meshwork and the juxtacanalicular meshwork. The corneoscleral meshwork is continuous posteriorly with the scleral spur, and the trabecular beams here are flat and have apertures which are smaller than the apertures in the uveal meshwork. The juxtacanalicular meshwork lines the inner aspect of the Schlemm's canal, and there are no trabecular beams here. Instead, the juxtacanalicular meshwork or the endothelial meshwork is made up of a multi-layered collection of cells loosely arranged in a ground substance, with the intercellular spaces between these cells not being more than 0.02 microns. And it is very important to remember that the juxtacanalicular meshwork is the site of the maximum outflow resistance. The Schlemm's canal is a circular tube along the circumference of the globe, and it is located external to the juxtacanalicular meshwork. And the Schlemm's canal is lined by a monolayer of endothelium with tight junctions between them with a discontinuous basement membrane. The inner portion of this lining lies adjacent to the juxtacanalicular meshwork and demonstrates pores and giant vacuoles. But the outer portion of the lining do not have any such pores, but do have exits of the collector channels. The endothelial cells have pinocytotic vesicles lined by membrane, and sometimes giant vacuoles are found which have openings which directly connect the extracellular space of the juxtacanalicular meshwork with the lumen of the Schlemm canal, thus forming a transcellular pathway for bulk flow of fluid. And as we have mentioned, these vacuoles are not found on the external wall of the Schlemm canal, and the size and number of these vacuoles increase when the intraocular pressure increases. 25 to 30 collector channels arise from the outer wall of the Schlemm canal and anastomose to form the deep scleral venous plexus and the intrascleral venous plexus. Aqueous veins, around 8 in number, arise from the intrascleral venous plexus and drain into the episcleral venous plexus, then to the anterior ciliary veins and the superior ophthalmic veins, and finally into the cavernous sinus. So we note here that the fluid draining through the trabecular meshwork is not collected by the vortex veins. These aqueous veins may be visible on the slit lamp with high magnification in the perilimbal area. Age-related changes occur in the trabecular meshwork which are accentuated in glaucoma patients, and these changes increase resistance to trabecular outflow. These include deposition of extracellular material in the extracellular matrix, resulting in thickening of the trabecular beams along with thickening of the basement membrane of the trabeculocytes. In addition, there is a decrease in the number of trabeculocytes, and in the remaining trabeculocytes, cross-linking of the actin microfilaments are found. There is also age-related increased pigmentation of the trabecular meshwork. Steroid therapy is known to cause glaucoma by increasing IOP, by increasing extracellular deposition of materials such as fibronectin, chondroitin, and proteoglycans, and these extracellular deposits on ultramicroscopic examination appear as fingerprint-like deposits. Steroids also increase IOP by increasing cross-linking of actin microfilaments within trabeculocytes. Steroid sensitivity of some normal individuals and glaucoma patients is said to be due to mutation in the glucocorticoid receptor GR4 beta. Rokinase or ROC inhibitors have been a recent introduction to the glaucoma armamentarium, and they are said to decrease IOP by disrupting cross-linked actin networks within trabeculocytes. Uveoscleral outflow, which occurs through the root of the iris and the anterior face of the ciliary body, contributes to as high as 50% of total aqueous humor drainage in young individuals, but decreases later. The fluid flow in uveoscleral outflow depends on a pressure gradient, but this pressure gradient is independent of IOP fluctuations in the normal range. And in glaucomatous individuals, there is increased extracellular matrix deposition in the constituents of this pathway. Prostaglandins improve uveoscleral outflow by stimulating ciliary muscle matrix metalloproteinase, thus stimulating degradation of these deposits. We note here that trabecular meshwork metalloproteinase is not stimulated by prostaglandins, and so prostaglandins have little effect on trabecular outflow. Several mechanisms have been proposed and evidence for them have been found to explain the death of retinal ganglion cells in glaucoma, and among them the first two are the most well accepted. Increased intraocular pressure causes compression of the retinal ganglion cells, and this prevents retrograde flow of trophic factors such as BDNF from the axon terminals of RGC to the cell body of RGC, which are essential for survival of RGCs. Vascular insufficiency may lead to chronic or intermittent hypoxic damage of RGCs, and other proposed mechanisms include oxidative stress, excitotoxicity from excessive glutamate, inflammatory cytokines such as TNF-alpha and nitric oxide, autoimmune response against retinal ganglion cells, and in addition to death of retinal ganglion cells, transsynaptic death of neurons in the lateral geniculate nucleus and the visual cortex. To recap the salient points, aqueous humor is produced from ciliary processes in the posterior chamber, passes through the pupil into the anterior chamber, and exits the eye from the angle of the anterior chamber. The ciliary processes are lined by two layers of epithelium, the non-pigmented epithelium adjacent to the anterior chamber and the pigmented epithelium adjacent to the ciliary stroma. across which aqueous humor is produced with the help of several channels, caryatids, and sodium-potassium ATPase pump. The sodium-potassium ATPase pump is present on the surface of the non-pigmented epithelial cell adjacent to the anterior chamber. Aqueous drainage can occur through the trabecular outflow route or the uveoscleral outflow route. Trabecular route consists of the trabecular meshwork, which in turn consists of uveal meshwork, corneoscleral meshwork, and the juxtacanalicular meshwork. Schlemm's canal, collector channels, aqueous veins, and episcleral veins. The juxtacanalicular meshwork is the site of the maximum resistance in the trabecular meshwork. Uveoscleral root consists of iris root, ciliary body, and supraciliary and suprachoroidal spaces. Uveoscleral root provides 50% of drainage in young individuals, but its contribution to drainage decreases later with age. Several mechanisms have been proposed for death of the retinal ganglion cell in glaucoma, among which IOP-induced and hypoperfusion-induced hypoxic damages are the most well accepted. Thank you for listening.